So, um, imagine you're standing right at the edge of this massive forest fire.
Like a completely devastating widespread fire.
Yeah, exactly. And, the firefighters arrive, they deploy all the hoses, and eventually those big visible flames die down.
Right, the smoke starts to clear.
Right. And on the surface, if you're just looking at the trees, it looks like a total success. The fire's out. But underneath the soil, like deep down in the peat bog, the fire actually hasn't gone out at all.
It's still smoldering down there.
Exactly. It's smoldering. And because it's underground, it's just slowly, quietly expanding outward, burning away the root systems of the entire forest. And this can happen for years, maybe even decades.
You don't see the raging flames anymore, but the forest just, well, it keeps dying.
And that terrifying imagery is actually incredibly precise when we talk about what is happening in the brain of someone living with progressive multiple sclerosis.
It really is. The visible surface-level flames of the disease might be gone, but that underground peat fire just continues to smolder and spread.
And that brings us to our mission for you today. We are going to dig into that metaphorical soil and find out exactly what is keeping that underground fire burning in the brain.
Because it's not just burning on its own, right? Something is actively fueling it.
Right. So we're unpacking this massive, genuinely groundbreaking study from Nature Neuroscience. And the researchers behind this paper have essentially identified the molecular sparks that drive this chronic, basically unstoppable destruction.
Yeah, and it's a very dense paper, but it's so important. By the time we finish this deep dive into the source material, you're going to understand the intricate turf wars of the brain's immune system.
And how, the aging process actually breaks down our cellular defenses. Plus how microscopic stress creates this vicious, self-sustaining loop.
Exactly. So the primary focus of this research centers on a specific group of highly toxic molecules called oxidized phosphatidylcholines.
There's a mouthful.
It really is. So the shorthand for them is OxPCs. We'll just call them OxPCs. The study maps out in just unprecedented detail how these toxic lipid byproducts drive chronic neurodegeneration through a very aggressive, deeply entrenched inflammatory pathway in the central nervous system.
Okay, but before we zoom in on all that molecular biology, I think it really helps to ground this in what a patient actually experiences. Because if you are familiar with multiple sclerosis, you probably first think of the relapsing remitting form.
Right, which is essentially that surface fire we were talking about.
Exactly. In that phase, rogue immune cells from the rest of the body actually manage to breach the blood-brain barrier. They invade, cause acute chaotic damage, and then they retreat.
Yeah, and the medical community has actually gotten quite good at fighting that specific type of fire.
Yeah.
Most of our current highly effective disease-modifying therapies target that exact mechanism.
They basically barricade the doors to the brain, right? So those peripheral immune cells just can't get inside.
Precisely. But the problem is, in progressive MS, the entire pathology shifts. We don't see those frequent leaky barrier invasions anymore. Instead, we see the rise of what neurologists call chronic active lesions, or CALs.
Okay, so if we were to look at one of these chronic active lesions under a powerful microscope, what does the geography of that battlefield actually look like?
Honestly, it looks like a scorched earth crater.
Wow.
Yeah, in the very center, the tissue is completely inactive and dead, just totally hollowed out. But the real danger lies at the perimeter.
The edge of the crater.
Right. Surrounding that dead core is this radially expanding rim of active destruction. At this outer edge, you find a swarm of highly reactive immune cells. You see the ongoing stripping away of myelin.
Which is the protective insulation wrapped around our nerve fibers, right?
Exactly. It's like the rubber casing on a wire.
Yeah.
And you also see the actual severing and death of the nerve fibers themselves.
So this expanding rim is the smoldering peat fire.
Yes. It slowly expands outward over months and years. And the scary part is its growth directly correlates with a patient experiencing worsening, irreversible physical disability.
So we know the fire is expanding, which means something has to be fueling it. It needs an energy source. And this is where the paper introduces those toxic OxPCs you mentioned.
Right, the oxidized lipids.
So walk us through how a normal, healthy piece of brain fat suddenly turns into this toxic fuel.
Well, it fundamentally comes down to oxidative stress. The human brain is incredibly rich in lipids, fats. They make up the structural membranes of every single cell up there.
Okay.
Specifically, a type of fat called phosphatidylcholine. Now, when brain cells are injured or suffocating or dying, They release reactive oxygen species as a byproduct.
Like exhaust from a struggling engine.
That's a great way to put it. Think of these reactive oxygen species as desperate, unstable chemical wrecking balls. When they crash into those healthy structural fats in the cell membrane, they oxidize them. They essentially rust them.
Oh, wow. So the physical structure of the cell is literally rusting, turning into this toxic OxPC byproduct. And the specific one in the study is called POVPC, right?
Yes, POVPC. And to figure out exactly how much damage this specific rust causes, the researchers couldn't just, watch a living human brain in real time. They had to build an entirely new animal model.
Right, because you can't just open up a human skull to watch this happen. So how did they build it?
They took a microscopic, highly concentrated drop of that specific OxPC, the POVPC, and they injected it directly into the white matter of the spinal cord in young 6-week-old mice.
Okay, I'm going to stop you right there because I have to challenge the premise of this setup.
Sure.
Injecting an artificial concentrated toxin directly into a mouse's spine seems like a really unnatural brute force way to study a human disease.
It sounds extreme.
Right. I mean, MS isn't caused by a scientist with a syringe. How can they possibly claim that a single injection in a rodent accurately mimics a complex human autoimmune disease that usually develops over decades?
That is a crucial question, and it's one the scientific community always asks when they're dealing with animal models. The researchers knew they had to prove this wasn't just a quirky reaction to a needle poke.
So how did they prove it?
To prove it, they used this revolutionary technique called spatial transcriptomics. SPRNA-seq for short.
Spatial transcriptomics. Break that down for us. What does that actually mean?
So imagine taking a tissue sample and creating a Google Earth map of its genetics.
Okay, I'm following.
You aren't just looking at what genes are turned on or off. You are mapping exactly where they are turned on geographically down to the absolute micrometer.
So you can see if a gene is active in the center of the crater versus the edge.
Exactly. The researchers mapped the genetic activity across the dead core and the expanding rim of the mouse lesion. And then they compared that exact geographical map to actual post-mortem brain tissue from human patients who had died with progressive MS.
Oh, wow. So they literally compared the blueprints of the destruction side by side.
And it was a nearly perfect match. The precise immune alarm bells ringing in the human brains, like specific complement genes and lipid processing genes, they were ringing in the exact same spatial patterns in the mouse brains.
That is incredible. So it proved beyond a doubt that the cellular environment created by that single injection of toxic lipid evolved into a true mirror of human progressive MS.
It completely validates the model. It's not just a fake injury. It's a real simulation of the disease state.
Okay, so knowing they have an accurate window into the disease now, what does the timeline of this destruction actually look like?
The persistence is what makes it so devastating. In most traditional lab models of MS, like EAE or LPC models, the mouse's brain eventually heals.
The fire goes out.
Right, the inflammation dies down, and the brain actually manages to rebuild the missing myelin. But in this OxPC model, the lesion simply refuses to heal, even at day 42 or day 100.
It just stays open.
Yeah. At day 7, which is the acute phase, it's absolute chaos. The nerve wires are visibly swollen, and the brain's local immune cells are massively enlarged and swarming the injection site.
Okay, so day 7 is the immediate 9/11 response. The sirens are blaring. But what happens when we fast forward to day 42? Because that's the chronic phase.
By day 42, the landscape is just desolate. The nerve wires in the core are entirely gone. The myelin insulation is virtually absent.
Total destruction.
But here is the real tragedy of it. The cellular electricians, these specialized builder cells called OLA-G2 positive cells, whose entire job is to rebuild that missing myelin insulation, they are actually sitting right there at the edge of the lesion.
Wait, really? They're on the construction site, but they aren't working.
Exactly. The local environment is so heavily saturated with toxic stress signals that the repair cells are essentially paralyzed. Remyelination completely fails, even though the builders are present.
That is so frustrating to visualize. But if I'm following the timeline correctly here, this brings up a massive biological mystery.
What's that?
Well, if the scientists only injected that tiny drop of toxic lipid on day 0, why is the environment still so toxic 6 weeks later? Wouldn't the mouse's body have just flushed that initial injection away almost immediately?
You have just hit on the absolute aha moment of the entire study. The researchers tracked that original injection, and they proved that the initial toxic lipid they injected was completely cleared from the spinal cord within 24 hours.
Wait, it's gone by day 1.
Gone by day 1.
But the damage is still actively expanding at day 42. How is that even possible?
Because the brain starts manufacturing its own poison.
Oh, wow.
Yeah. The initial injection triggered a localized wave of cell death and oxidative stress. And that stress released those chemical wrecking balls we talked about earlier.
The reactive oxygen species.
Right, which then crashed into the surrounding healthy lipids, oxidizing them and creating brand new endogenous OxPCs.
It's a chain reaction. The smoldering fire is generating its own fuel. So the rusting of one cell spreads the rust to the neighbor, which spreads it to the next neighbor.
Precisely. And they prove this using a specialized fluorescent antibody called EO6 that only binds to these oxidized lipids. When they applied it to the tissue at day 42, the entire rim of the lesion just lit up brilliantly.
So the original toxin is gone, but the brain is now saturated with its own auto-generated toxins.
Exactly.
Okay, let's pause and synthesize what we've uncovered so far, because this is dense and it fundamentally shifts how we view the disease. We'll do a quick 5-minute recap.
Sounds good.
I want to use a mnemonic here for the listeners. Think of the word CALM, C-A-L-M.
I like that.
C is for chronic active lesions. A is for auto-generating lipids, which are those OxPCs. And LM is for mimicking human PMS.
That perfectly summarizes the first part of this paper.
So bullet point summary. 1, OxPCs cause these chronic active lesions. 2, the brain eventually starts making its own OxPCs in a chain reaction. And 3, because of this toxic environment, remyelination totally fails. The builder cells are paralyzed.
Exactly. The initial spark is long gone, but the fire is fueling itself.
Which brings up a really critical question for you, the listener, to consider. If your brain suddenly started manufacturing a spreading toxic poison, what cellular first responders would you want on the scene? Let's look at the cells responding to this fire. Who exactly answers that 911 call, and why are they failing to stop it?
To understand why the response fails, we really have to look closely at the cellular turf war happening at the very edge of the lesion.
A turf war.
Yeah, because there are essentially 2 different populations of immune cells fighting this fire. First, you have the microglia. These are the brain's resident, lifelong protectors. They are born in the brain and they stay in the brain.
So they are the local fire department.
Yes. And the second group are the monocyte-derived macrophages, or monomacs for short. We can call them the out-of-towners.
Okay, the out-of-towners.
These are heavy-duty, aggressive immune cells that normally circulate in the bloodstream. They only cross over into the brain when there is a massive emergency.
So it's the local firefighters versus the federal SWAT team.
Exactly. And the researchers noticed a very specific identity crisis happening over time in the mouse models. In the acute phase, right after the injury, the local microglia are everywhere just trying to manage the damage.
And there's jobs.
Right. But as the lesion turns chronic and expands toward day 42, those out-of-town monomacs increasingly invade the brain and actually begin to replace the local microglia at the lesion edge.
Wait, so the local firefighters are just disappearing and simultaneously the lesion is getting worse and expanding?
Yes, exactly.
Well, that correlation makes me wonder about causation. Microglia are inflammatory cells, right? When they activate, they cause collateral damage. So could it be that the local microglia are actually the arsonists here? Are they the ones driving the expansion of the lesion?
That was the exact hypothesis the scientists needed to test. And to find out, they designed an incredibly elegant, highly targeted depletion experiment. They used mice that were genetically engineered. They're called CX3CR1 Creer mice. But essentially, they had a hidden vulnerability built exclusively into their microglia. Think of it as a remote-controlled kill switch.
A kill switch that only affects the local firefighters, completely ignoring every other cell in the brain.
Correct. By giving the mice a specific combination of drugs, including diphtheria toxin, they could flip that switch and trigger the microglia to undergo apoptosis.
Meaning they just quietly died off.
Yes. So, the scientists let the toxic lesion develop normally. Then, right in the middle of the chronic phase, when the fire was burning underground, they flipped the switch and wiped out the local microglia.
They intentionally removed the firefighters to see what the fire would do.
Exactly.
So what happened to the brain?
The pathology went completely off a cliff.
Oh, wow.
Yeah, without the local microglia, the accumulation of those toxic rusted lipids absolutely skyrocketed. The destruction of the nerve axons got significantly worse, and the dead core expanded much faster.
So they weren't the arsonists at all?
Not at all. It proved definitively that the resident microglia are the good guys. They are desperately trying to scavenge, engulf, and clear away the auto-generating toxins to protect the surrounding healthy tissue.
That paints such a tragic picture, honestly. The microglia are essentially throwing their bodies on the fire, trying to absorb the toxins, but they're just getting overwhelmed and replaced as the disease drags on.
Right, they just can't keep up.
But there is another variable here that is universally relevant to human patients. Progressive MS almost always worsens as a patient gets older. It is a disease heavily intertwined with time. Did they factor aging into this model?
They did, and the results were really stark. They repeated the whole experiment, but this time they compared young 6-week-old mice to middle-aged 52-week-old mice.
What did they find?
In the older brains, the chronic lesions were vastly larger. The destruction was far more severe.
How do they actually measure the severity of nerve destruction specifically? I mean, it's not like you can ask the mouse how it feels.
Right, no. They look for a specific molecular distress signal. It's a marker called beta-APP.
Beta-APP, okay, how does that work?
Imagine a healthy nerve fiber as a busy highway, right? Constantly transporting cellular cargo up and down its length. When that nerve fiber is severely injured and starts choking, the highway gets blocked.
So the cargo has nowhere to go.
Exactly. The cargo violently piles up at the site of the damage, and that piled up cargo is beta-APP. In the older mice, the accumulation of beta-APP was massive. It looked like a catastrophic multi-car pileup of dead and dying nerves.
That's a great analogy. So why does the older brain fare so much worse? What is actually changing in a 52-week-old brain compared to a young one?
It connects directly back to our local firefighters.
Yeah.
As the brain ages, the microglia become senescent. They literally grow old and tired.
Oh, they age too?
Yeah, their physical ability to scavenge and swallow up those toxic lipids just breaks down over time. So the firefighters are aging, their equipment is failing, and without them constantly clearing the brush, the smoldering peat fire just burns out of control.
Okay, let's gather these threads together for our next quick recap.
Good idea.
Bullet summary time. 1, we've established the local microglia are the crucial defenders, the good guys trying to clean up the toxic lipid spills. 2, we see that as the disease progresses, the aggressive out-of-town immune cells, the monomacs, take over the territory.
Right.
And 3, we know that aging is a massive risk factor because it cripples the microglia's ability to fight back, leading to catastrophic nerve death.
Exactly.
Which brings us to the most important question of the entire deep dive. We know the older brain struggles to put out the fire, but what exactly is the fuel keeping that fire burning? What is the actual chemical command, the literal molecular engine that forces this chain reaction to keep running in a loop?
To find that engine, the scientists had to look past the dismal inflammation and identify a specific molecular culprit. And they found it in a tiny but remarkably potent inflammatory protein.
What's it called?
Interleukin-1 beta, or IL-1 for short.
IL-1. Okay, to understand how dangerous this specific protein is, you really have to visualize it as an audio feedback loop.
Oh, that's a good way to look at it.
Yeah, if you've ever been at an event where someone holds a microphone just a little too close to the speaker, you know exactly what happens. A tiny bit of background sound goes into the microphone. It comes out of the speaker louder. That louder sound goes right back into the microphone, comes out even louder, and within seconds you have this screeching, deafening loop that just blows out the whole system.
That is the perfect analogy for what IL-1 does inside a chronic lesion.
How so?
Well, here is how the loop functions chemically. The toxic OxPCs sitting in the tissue irritate the immune cells. In response, those immune cells release IL-1.
Okay, so the protein is out there.
Right, and the IL-1 then triggers intense localized inflammation and massive oxidative stress.
And as we learned earlier, oxidative stress acts as a wrecking ball that rusts healthy fats.
Exactly. The IL-1-induced stress rusts the neighboring healthy lipids, which generates a fresh batch of toxic OxPCs. And then those new OxPCs irritate the immune cells again, causing them to release even more IL-1. The microphone gets closer and closer to the speaker, and the biological volume just keeps increasing.
It is a vicious, completely self-sustaining chemical loop. Logically, then, if you could just snip the microphone cord, like if you could somehow stop the IL-1 from functioning, you could break the loop, stop the generation of the toxic lipids, and save the surrounding brain tissue.
And that was the ultimate hypothesis of the paper. And they proved it beautifully using a genetic knockout model.
How do they do that?
They bred a unique lineage of mice that lacked the genetic instructions to build specific enzymes called caspases, specifically caspase 1 and 4.
What exactly does a caspase do in this scenario?
Think of caspases as highly specific molecular scissors.
Okay, scissors.
When a cell decides to manufacture IL-1, it doesn't build it in its active, dangerous form. That would be way too risky. It builds it in a locked, inactive state called pro-IL-1.
So it's safe at first.
Right. It is completely harmless until the caspase scissors come along and literally snip off the biological padlock, activating the protein.
Ah, I see. So by breeding mice that simply didn't have the scissors, the researchers guaranteed that even if the cells tried to make IL-1, it would remain permanently locked and inactive.
Precisely. They took these scissorless mice, injected the initial drop of toxic lipid to start the fire, and waited 32 days.
And what happened? Did the loop break?
The results were incredible. Without the active IL-1 to drive the feedback loop, the accumulation of new endogenous OxPCs was dramatically reduced. The vicious cycle never took off.
That's huge.
And most importantly, the nerve wires the axons they were overwhelmingly spared. By genetically neutralizing just one specific protein, they halted the chronic neurodegeneration.
That is a phenomenal proof of concept in a laboratory.
Yeah.
But obviously, you cannot gene edit the entire immune system of a human patient who is already sitting in a neurology clinic with progressive MS.
Yeah, you can't.
I mean, the listener is likely thinking, that's great for the mice, but can we actually block this loop in a living, normal brain using medicine?
We can. And the most exciting part is that the medical community already has the tool. It is a drug called anakinra.
Anakinra. How does a drug accomplish what genetic engineering did?
Anakinra acts as an IL-1 receptor antagonist.
Okay, what does that mean in plain English?
Well, it doesn't stop the cells from manufacturing the IL-1, and it doesn't act like the molecular scissors. Instead, it physically blocks the cellular receptor that IL-1 tries to plug into.
Okay, let me try an analogy. If IL-1 is a key trying to unlock widespread inflammation, anakinra is the glue that fills up the keyhole.
That's exactly it. The key still exists, but it just can't turn the lock.
So the researchers let the underground fire start in normal, non-engineered mice. They let it burn until it reached the chronic phase, day 28 to 42, where the damage is usually permanent, and then they intervened with anakinra.
Yes.
What happened?
Intervening with the drug halted the progression of the disease in its tracks. By covering the keyholes from day 28 to day 42, anakinra significantly reduced the volume of toxic oxidized lipids in the brain. It successfully shielded the nerve fibers from dying.
That is amazing.
And in a truly fascinating twist, cutting that inflammatory loop actually promoted the return of the healthy, homeostatic local microglia to the lesion site.
Oh, wow. So it didn't just act as a fire extinguisher, it actually cleared the smoke so the original local firefighters could return to the scene and do their jobs properly.
Exactly. They came back.
That is the kind of biological elegance that gives you genuine hope. And this isn't just a theoretical concept sitting on a laboratory bench, is it?
Not at all. The translation from this specific research to clinical application is moving rapidly. The mechanistic connection is so strong that there is currently an ongoing clinical trial testing anakinra specifically for chronically inflamed, non-healing white matter lesions in human multiple sclerosis.
Seriously.
Yes. They are actively trying to jam this feedback loop in human patients right now.
That is incredible. Okay, let's synthesize this final piece of the puzzle. Time for our last recap.
Let's do it.
So, bullet one. We discovered that the entire architecture of chronic brain destruction in this model relies on a screeching feedback loop between rusted lipids and the inflammatory protein IL-1.
Correct.
Bullet two. We saw that if you genetically remove the molecular scissors, the caspases, that activate that protein, you save the nerve fibers.
Right.
And bullet three. We learned that using an existing pharmacological blocker like anakinra covers the cellular keyholes, cuts the feedback loop, and actually clears the toxins.
Perfectly summarized.
Which brings us to the ultimate question for you to ponder. We've seen exactly how cutting this specific feedback loop can save the brain in a mouse model. But what does this fundamentally mean for the future of how we approach and treat all neurodegenerative diseases?
If we pull all the way back and look at the entirety of what we've unpacked today, a profound paradigm shift emerges here. For decades, the persistence of progressive MS was medically baffling. It was largely viewed as just the slow, inevitable, untreatable scarring from past immune attacks.
Just a scar you had to live with.
Exactly. But this deep dive reveals that it is not passive scarring. It is an active, ongoing, highly specific chemical loop. It is driven by a precise mechanism, IL-1 and oxidized lipids, and compounded by the failing clearance of aging immune cells.
It transforms the disease from an unstoppable ghost into a physical machine. And once you have the blueprints to a machine, you can figure out how to break it.
Right. Whether it is using advanced antibodies, like that EO6 we mentioned, to act as a targeted sponge to soak up the toxic lipids, or using receptor blockers like anakinra to jam the gears of the inflammation loop, we now have real, tangible, biological targets. It provides a massive, biologically validated rationale for hope. It proves that chronic neurodegeneration is not an empty void we simply have to accept. It is a highly ordered biochemical pathway that can be forcefully interrupted.
I want to leave you with one final kind of provocative thought to mull over long after this deep dive ends.
Oh, what's that?
Well, the study notes a very specific universal biological rule. Whenever cells undergo severe stress and die, they naturally generate these oxidized lipids. It is a fundamental byproduct of cellular distress.
That's true.
So if our brains naturally produce these toxic rusted fats under high stress, could this exact same IL-1 feedback loop be the hidden driver behind entirely different forms of age-related cognitive decline, far beyond the specific diagnosis of multiple sclerosis?
That is a fascinating question.
Right. Could it be playing a role in Alzheimer's or Parkinson's? Think about how your own daily chronic stressors, the cortisol spikes, the lack of sleep, the systemic inflammation from diet, how that might be leaving an invisible accumulating lipid footprint in your own brain over the years.
It absolutely forces you to reevaluate cellular stress through a much wider lens, because the fundamental mechanisms of lipid rusting and inflammatory feedback are not unique to MS. They are universal features of how the human body ages and responds to injury.
Exactly. The implications are basically boundless. Thank you for joining us on this deep dive into the source material. We started today by staring at a smoldering peat fire, watching it quietly destroy the roots of a forest from below. But by digging into the soil, we didn't just find the hidden embers. We mapped the exact chemical reaction keeping them dangerously hot, and we found the extinguisher. Keep questioning the world around you, keep learning, and we'll see you on the next exploration.